Introducing mitosis, meiosis, and mutations.
Mitosis duplicates DNA into two daughter cells with an exact genetic code.
Meiosis is a division in a cell into 4 haploid cells with non-identical chromosomes.
Mutations are changes in the genetic material of a cell or virus that can occur in mitosis or meiosis. Mutations can be caused by mutagens, which are physical or chemical agents.
First, we can look at a point mutation, which are chemical changes in just one base pair of a gene. Point mutations can be broken down into 2 categories known as base pair substitutions and base pair insertions or deletions.
Base pair substitution replaces one nucleotide and its partner with another pair of nucleotides.
Silent, missense, and nonsense mutations also fall under the base substitution category.
Silent mutations have no effect on the amino acid. For example, when codon AAA turns to AAG, the amino acid produced is still the same, therefore causing no effect.
Missense mutations occur when new nucleotides alter the code and produce alternate amino acids.
For example, sickle cell anemia is the replacement of A with T at the 17th nucleotide and becomes valine instead of glutamic acid.
A nonsense mutation changes an amino acid codon into a stop codon, almost always leading to a non-functional protein. Insertion is when an extra base pair may be added, like with Huntington's disease. In contrast, deletion is when that extra base pair may be removed, such as with the cleft palate. Insertion or deletion can change the frame, causing a frame shift, and the gene cannot be coded correctly, like with Crohn's disease.
Each cyclin is associated with a particular phase, transition, or set of phases in the cell cycle and helps drive the events of that phase or period.
For example, the M cyclin promotes the events of M phase, such as nuclear envelope breakdown and chromosome condensation.
Cyclin-dependent kinases, also known as CDKs, are a family of enzymes that become activated by binding with cyclin, allowing them to modify target proteins.
Maturation promoting factor is a cyclin CDK complex that triggers a cell's passage past the G2 checkpoint into the M phase.
Oncogenes are cancer-causing genes. Cancer can be caused by mutations to genes that regulate cell growth and division.
Opposite of oncogenes are proto-oncogenes, which are the corresponding normal cellular genes responsible for normal cell growth and division.
Mutations in the RAS gene can lead to production of a hyperactive RAS protein and increased cell division.
Mutations in the p53 gene prevent suppression of the cell cycle.
Non-disjunction is when a pair of homologous chromosomes do not separate normally during meiosis. The result is that one gamete receives 2 of the same type of chromosome and another gamete receives no copy.
Aneuploidy results from the fertilization of gametes in which non-disjunction occurs. Offspring with this condition have an abnormal number of a particular chromosome.
Monosomic is when a zygote has only one copy of a particular gene.
A trisomic zygote has 3 copies of a particular chromosome.
Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. It's common in plants but not in animals.